Okay, this is part 2 of tissues. What we're going to be doing in this part is just taking a look at the classifications of simple types of epithelial tissue. So just remember, simple tells us that it is one layer. Squamous is a flattened cell, just as a refresher. So it shouldn't be a surprise then that the functions we're going to see associated with simple squamous is going to be diffusion and filtration, where we want something that is incredibly thin. But if we wanted some sort of protection, let's say, we're going to want an incredibly thick layer and not something that is really thin. So it's going to try and reduce friction in the lymphatic and cardiovascular systems, and we're going to see it now. The textbook is going to give you much more locations. I'm trying to kind of narrow it down. So the kidney glomerulus and blood vessels, because that's where you're really going to be seeing them in lab. In blood vessels, though, this is noteworthy, literally, endothelium is a special name, and that tells you that it is simple squamous lining blood vessels. If we have simple squamous lining something else, mesothelium is going to be found, it's simple squamous lining various mesenteries. But by far, endothelium or an endothelial layer, you really need to know what that is. I'm not going to spend too much time on these images because that's what lab is going to be for, either seated or online. But nonetheless, just super quick. See, this is one thin layer, and these black dots here, those are going to be the nuclei. Okay. Next we have is the simple cuboidal, and that's going to function in secretion and absorption, and we're going to see that also in the kidneys and the secretory portion of small glands. Really, where you're going to see this in lab is the thyroid gland. You don't need to know that for lecture. It's just that's what you're actually going to be taking a look at. This image is just showing us a cross-section of the kidneys. These are all of these tubes cut in cross-section. So you're kind of looking at like a manhole cover. And again, not sure if I mentioned this in the previous one, but because epithelial tissue is so jam-packed full of cells, we're not going to always be able to see the where one cell ends and another one begins. So that's where using the nucleus really helps. So you can see from this apical surface here to this basal surface here is one cell, the spherical nuclei, that is simple cuboidal. Simple columnar. Goblet cells are typically going to be found in this layer. Goblet cells secrete mucus. Okay. Function, absorption, and secretion. We have two different types, a non-ciliated type and a ciliated type. Non-ciliated we're going to see in the digestive tract. Ciliated type in the small bronchi and the uterine tubes. So here they're showing simple columnar. This is in the digestive system. And again, in the previous one, I said there's one example where we have a columnar cell with a circular nuclei, but still it should be pretty obvious that that is going to be a columnar shaped cell. All right, last one, pseudostratified columnar epithelium. Pseudo, that prefix means false. So it looks like it's stratified, but it really is not. So it's a single layer of cells with different heights. Some do not reach the free surface. So when we take a look at this, what we're going to see is that nuclei are seen at different layers. I would attempt to draw this, but I really would make it worse. So function and secretion and propulsion of mucus, and present in the trachea and the male sperm carrying ducts. The ductus deferens. The examples that we have in lab, I believe, are the trachea. And just as an FYI, if you're in a seated part, you can also see hyaline cartilage there as well. So we're going to take a look at this image. Now, this is one of the few times where the image that they show you isn't that great. First of all, we have all of this mucus around these goblet cells that are kind of interfering with our ability. If you take a look at Blackboard, I've put up a much better example. So if you're kind of confused being able to see that this right here is pseudostratified columnar, don't worry. This is really what I wanted to show you, where you can see that the nuclei are seen at different heights, but it is not a stratified layer. We don't have one cell on top of another. All right. So all of these cells are either going to be attached to the basal surface or the apical, but it is not a truly stratified layer. Again, don't worry too much about this example. I have a much better example on Blackboard. Next one, we're going to be taking a look at the stratified layers.